Hi guys, this is Dr. Momsilovich with your lecture 5.2 on microbial eukaryotes. Last lecture we discussed some of the simple single-celled organisms, the prokaryotes, which are the bacteria, and the archaea. This lecture and the next couple of lectures we're going to be talking about eukaryotes, which are a little bit more complex forms of life. We go back to our tree of life where we were discussing our domains. The last time we talked about the domain bacteria and the domain archaea. Now remember, those were all single-celled, very simple, no nucleus, all of that kind of stuff. Now we're going to be talking about the eukaryotes or the domain eukarya. This includes some single-celled organisms that are a little bit more complex in how they're built, as well as multicellular organisms. So the difference between a prokaryote and a eukaryote is essentially the complexity of the cell type in those organisms. So you remember a prokaryote has a nucleoid instead of a complete nucleus with a cell membrane. It has ribosomes, which eukaryotes also have. It has, prokaryotes have cell membranes, which eukaryotes have. Prokaryotes have a cell wall and a capsule, which eukaryotes don't have. And then prokaryotes can have a flagellum, which eukaryotes do not have. Instead, eukaryotes have several other organelles that have arisen. And so this question becomes, if the first cells were simple, like modern prokaryotes, then how did complex eukaryotic cells arise? And our best hypothesis for this, and what is actually no longer a hypothesis, now it's a theory, is the theory of endosymbiosis. So, on the last slide we talked a little bit about what prokaryotic cells and eukaryotic cells have that are different. In general, things that eukaryotic cells have that prokaryotic cells don't have is an endomembrane system, which, if you guys remember, means that they have rough endoplasmic reticulum, smooth endoplasmic reticulum, they have Golgi apparatus, and they have lysosomes. That's all part of the endomembrane system. They also have a nuclear envelope, which is the surrounding membrane around their nucleus that keeps the DNA in a central location. They have mitochondria, which is something that all eukaryotes have, and they have chloroplasts, or some of them do if they're plants or if they do photosynthesis as a single-celled organism. So a scientist named Lynn Margulis is the first one to propose endosymbiosis as a hypothesis for how prokaryotic cells evolved into eukaryotic cells. And she did this back in 1966. And she said that the mitochondria, the photosynthetic plastids, and the 9+2 basal bodies of flagella were themselves once free-living prokaryotic cells. And so this idea of endosymbiosis, if you think about the roots of that word, endo meaning internal, and we know symbiosis is a mutualistic relationship between two organisms, it's this idea that eukaryotic cells evolved from prokaryotic cells by taking in other cells and creating a symbiotic relationship with them while they were stored inside of their own cell membrane. So one question that comes from this is, where did our nucleus and our endomembrane system come from? And it's generally thought that the outer cell membrane invaginated to form the nuclear envelope and the endomembrane system. This is what we call the outside-in model. So invaginate means that the outer cell wall folded in on itself and therefore created a double membrane inside the cell. And that is where our endomembranes came from in more complex cells. So the rest of the endosymbiotic theory explains how cells were able to evolve to have mitochondria and chloroplasts. So remember that the first step of this was that you had an infolding of the plasma membrane of an ancestral prokaryote, and that gave rise to the endomembrane components, including a nucleus and an endoplasmic reticulum. From there, these ancestral cells were then able to have endosymbiotic events through the process of phagocytosis, where they took in smaller bacterial cells, and that those cells became our mitochondria and the chloroplasts in plants. And so when a cell does phagocytosis, it essentially sucks another cell or something else into its membrane. And so the first one we think was when a cell consumed an aerobic bacteria that evolved into mitochondria. The second event would have been when another cell did the same thing, phagocytosis, and took in a photosynthetic bacterium, and that created a modern photosynthetic eukaryote. So, the next question is, do we have any evidence for this process of endosymbiosis occurring in early prokaryotes, turning them into eukaryotes? And we actually do. We have quite a bit of evidence, and that evidence has turned this hypothesis of endosymbiosis into a theory instead. So the first piece of evidence that we have is that mitochondria and chloroplasts have their own genomes, and it's a circular chromosome like that we see in free-living bacteria. So over time, the genome in the, mitochondria and chloroplasts have gotten smaller because as they have been inside of another cell, there are things that the cell does, and therefore proteins that are made and things like that, that those structures don't need to do anymore. And so their DNA has actually gotten smaller and a little bit simpler, but they do still have their own DNA. They're not coded for by the cell's DNA. The second evidence is that mitochondria and chloroplasts both replicate via binary fission like bacteria do. So when we think about what that could mean, you only will inherit, for the most part, and very rarely does it go any other way, but you in general will inherit all of your mitochondria from your mother's egg cells. You don't inherit your mitochondria from your father's cells, and it's because in those egg cells they have mitochondria, and those divide by binary fission and get put into all of her egg cells. There's not mitochondria generally in sperm cells, and so therefore you're not inheriting it from your father. And so you can actually trace your ancestral heritage. One way you can do it is through looking at your mitochondrial DNA, and so you can consistently trace back your maternal heritage to see where all of that mitochondria came from and move back that way, because you're only inheriting that from your mother. The third line of evidence is that both organelles are surrounded by a double membrane, and that was acquired by ingestion by the ancestral host. And so they have the same double membrane that you would find in a free-living prokaryote. What's really fun and really interesting about this idea of endosymbiosis is that we actually have modern examples of the initial stages of endosymbiosis that we can see living today. So when you think about it, generally a cell would use the process of phagocytosis to take something in that it's going to then dissolve and use for its own food. However, we know that all cells don't necessarily do that, and so they can actually create a symbiotic relationship, and therefore they're able to continue operating in that same way. So one example is in a paramecium, and these are simple cells that will ingest cyanobacteria, and they don't consume it. They just keep it within their body cell, and the paramecium and the cyanobacteria create a symbiotic relationship where the paramecium is giving the cyanobacteria a safe place to live, and it moves the cyanobacteria to areas with lots of sunlight, and therefore they can do photosynthesis and create glucose and other sugars. And in return, the cyanobacteria share that sugar that they're creating through photosynthesis with the paramecium. And so they have a symbiotic relationship. The other example that we have, or one of the other examples, is in sea slugs that keep algae in their system, and then they use, in a similar way to the paramecium, they use the sugar that's generated when the algae do photosynthesis. And so the sea slugs will take in the algae and put it into their tissues, and then the sea slug can move to areas with lots of sunlight and allow that that algae to do photosynthesis and create sugars in the same way that the paramecium moves around for the cyanobacteria. Within the domain of eukarya, there are 6 eukaryotic supergroups. We're going to talk about 4 of those in more detail today, and then 3 of them are going to be in more detail in the next couple of lectures. So the ones we're going to talk about in the future are the land plants, fungi, and animals. And those 3 lineages are unique and important because they evolved multicellularity independently of each other 3 separate times. The remaining groups are primarily unicellular or colonial and are often called protists. So in high school, you probably learned that there was the domain Protista, and we now group protists as part of the domain Eukarya. The first supergroup that we're going to discuss within the domain of Eukarya is the Archaeplastida. These all descended from an endosymbiotic relationship between a heterotrophic protist and a cyanobacterium. There are 3 different types within this supergroup, and the first one are the glaucophytes. The second one are red algae, which are unique because they have a pigment called phycoerythrin that gives them their reddish color. And then there is the green algae, which is the algae that you are probably most familiar with that grows in your pool or your fish tank or your nearby lake. The next supergroup is the amoebozoa. These consist of large cells with multiple nuclei, and they move via cytoplasmic streaming in their pseudopodia. So this group you probably looked at in your high school biology course, and you can remember them sort of crawling along the surface. So they'll extend out their pseudopodia and then sort of pull the rest of their body along with them. These eukaryotes feed via phagocytosis, and they have clues to the evolution of multicellularity from the slime molds, which are shown in the time-lapse video down in the bottom right-hand corner of this slide, which move as a colony, and they coordinate their movement to find food and to digest it. And so we think that they are sort of the basis of multicellularity. The next supergroup is the Epistheconta. So it's actually kind of interesting to think that you are an Epistheconta. These are cells that have a single posterior flagellum. So if you think about what a sperm cell looks like, it looks very similar to Epistheconta. The next group is called the Epistheconta. It's kind of fun to think about the fact that you were, at one point in your development, an Epistheconta or very similar to it. Their structure is a single-celled organism with a cone around the base of their flagellum, similar to what a sperm looks like. We call these choanoflagellates, and they are the sister group to the animals. So at the bottom of your slide, you have a picture of a choanoflagellate, which is a protist, and then you have also what the choanocytes in a sponge, which are the basal animals, they have choanocyte cells in their structures, and they are the exact same structure as a choanoflagellate. So we can sort of see the transition from being a single-celled choanoflagellate to a multicellular sponge that's made partly of choanocyte cells in the earliest animals. The next group are called the Rhizaria. These have thin thread-like pseudopodia and are actually kind of pretty. They have outer shells that are made from calcium carbonate or silicon. They have, there's 3 groups within them. There are foraminiferans, struggled on that one. These are common in marine benthos, which are the ecosystems at the very bottom of the ocean. There are the radiolarans, which are common marine plankton, and the cercozoans, which we find often in soil. The next group are called the chrome alveolata. These are actually kind of a fun group. So they evolved from a common ancestor that underwent secondary endosymbiosis. So they ended up not only taking in a chloroplast from a photosynthetic prokaryote, but also a photosynthetic red algae. There are 5 different types of chromia alveolata. The first one are the dinoflagellates. These occur in fresh and marine plankton, and these, if you've ever seen the algae that bioluminesce, that's what these guys are. The next group within this are the apocomplexans. These are Plasmodium. Plasmodium is a single-celled eukaryote that causes malaria. There are ciliates, which are paramecium. We talked about paramecium and that they used phagocytosis to take in cyanobacteria and have an endosymbiotic relationship with them. We also consider diatoms to be part of this group. That's another one you may have talked about in high school. There's diatomaceous soil, which we use to get rid of insects and things like that. And this group also includes brown and golden algae. So this one's a pretty diverse group that includes several different types of algae that range from bioluminescent to golden. It includes paramecium. This one's pretty fun and pretty charismatic. Last group are called the excavata. These are asymmetrical, which is kind of unique, and they are single-celled organisms. And most of them are parasitic, and there's a variety of different parasites that fall into this group. One of them is Giardia, which you may have heard about if you like to hike or camp or you're outdoorsy. And it's a waterborne eukaryote that causes issues with your GI or your gastrointestinal tract. And so these guys are the reason that you don't want to drink stream water or pond water or anything like that without purifying it first. They can cause a lot of stomach issues for you. Another one are the trichomonas, and these ones cause an STI, so it's sexually transmitted infection that can cause some pain and some oozing and some other things in your pelvic area. And then the last one are the trypanosoma, which cause sleeping sickness, and they are transmitted by the tsetse fly. So that ends our discussion of the simple eukarya, and in the next lectures we're going to talk about basal plants, fungi, and the animal kingdom.